start again, chapter 3 of the module 6. Genetic technology is basically something that we continuation of something we learned like in the 2 weeks ago. That's a biotechnologies. So going back to the biotech and the biotechnologies again, biotechnologies. So biotechnology is the kind of bigger terms in terms of like describing the and this contains a lot of the different type of the technology involving some living organisms. So a good example of the old-fashioned like biotechnology, we discussed that the fermentations and whatnot. We're using the yeast or the bacteria for the fermenting the foods or the fermenting some other purposes. The herbal medicine, it's also the biotechnology. We discussed that too. Okay. Yes. Now, also among the lots of the you're living in comedy, okay?
Oh, so it's alright.
Okay, so among the biotechnology, genetic technology is quite a recent technology because it's been like only like few 10, 20, 30 years that we're developing those. So which means that we are still developing these technology now, even now. So you probably haven't heard about, we kind of bring up some of the ethical issues using the biotechnologies, like GMOs, the genetic modified organism or the foods. Everyone got shocked that salmon was a part of the GMO foods nowadays, okay? But if you think about all the foods that you're consuming, like the sugars, for example, they're all basically genetically modified. So that is the one example of the gene technologies. So among that, the biotechnology, we have the genetic technology. So genetic technology means that we kind of narrow down the biotechnology into something that involved with the genes, especially about the genes. We got to change that. Now, to understand the genetic technology, it's good to start off with the term called the gene then. In module 5, or like probably in the year 10 and year 11, you learned that gene is the section of the DNA. And we learned that as because the section of DNA is corresponding to responsible or corresponding to very special features. Changing a little bit of the section of the DNA, they can cause they call the mutation. That's something we learned in chapter 1. So this is how you need to collaborate and working collaboratively from the module 5 and 6, and thinking about that too. Now, gene is also be corresponding to something that we learned in module 5, the polypeptide. The synthesis. Changing the structure of the gene or the sequence of the base pairing, basically that's you get the very different features of the function of the DNA, so the proteins. If that's your enzyme, it's hormones, you get a completely different functions too. Sometimes we kind of design that, we kind of modify that to get the certain desired effect. That's also the part of the biotechnology then. Among that genetic technology, we have many different types. One type of the genetic technology is known as the reproductive technologies. As you can get from the title reproductive technology, this is something that related to all the reproduction. So I'm talking about sexual reproduction of the eukaryotics, specifically for like all the some of the organisms of the foods. So a good example, as we learned, is the selective breeding. It is one example of the reproductive technologies. What is selective breeding? It's a good example. It is also the same as like an artificial, like a selection that too. We chose that very certain phenotype of the organism, like puppies. If you're looking for like a small, cute little puppies, we're only going for like those puppies. And then we're just kind of crossbreeding with this similar types, and the similar phenotypes. You only get the small puppies nowadays for that reason too. So, selective breeding in animals highlight that place in the most basic form involves mating a male that displayed at least one desirable characteristic with the female that displays at least another desirable characteristic, in the hope that some offspring will inherit the desired favorable genetic traits from the both parents. So for example, cross A, Frisian and Frisian bull. The female Frisian are known for their production of large quantities of milk, and a Jersey cow, which has produced the creamy milk, will result in some offspring that produce large amount of the creamy milk. This is basically what we get nowadays, because back in the many hundred years ago, milk wasn't that creamy. Milk wasn't that kind of fatty. It was kind of yeah. But nowadays, kind of by selectively choosing those and mating those, we're now able to get those creamy milk. Or good example, if you remember that Wangoose. Wangoose. Yeah, that's a good example of the. I don't know like if we'll actually get the Wangoose because I've never seen that in the market yet, but that's probably one of the things you can think about the crossbreeding too. We only get the benefits of the Wagyu and the Angus. Yummy. Yes, that's all we want from that. All right, some disadvantages here. This is kind of random, by the way. This is random, which means that you sometimes get the desired phenotypes, sometimes you don't. Okay, depends on because all as we learn the sexual reproduction, it happens kind of randomly. So offsprings that have both of these traits are known selective that blah blah blah. Okay, second dot point, please. Another disadvantage of using whole animals for the selective breeding is that it is time-consuming and costly too. Indeed, it takes a lot of time. Think about like how long does it take, like the time taken for raise one cow into adult cow. And if that failed, you need to start it again. If it takes like 10 years to raise it up to 5 years to raise up the cow, I mean, if you failed one generation, you gotta start it again, going for the next generation. That takes a super amount of time and the cost as well too. It involves that transporting large animals over long distances. There is always chance that they will injure each other and that may or may not mate, or that will take a long time for the cow to become pregnant too. So, to overcome, highlight this one too, the technology of the artificial inseminations, in vitro fertilization, in short IVF, or the multiple ovulation embryo transfer, we call the MOET, have been introduced from it. So this is actually like where we're thinking about genetic technology from. So a lot of time genetic technology is overcoming those disadvantages of the previous successes, which is like they are costly, they are time delaying. So a good example is the artificial insemination. Please highlight the first dot point there. Artificial insemination in animal involves collecting sperms from their chosen male and artificially introducing it into the several selected females, rather than we waiting for their naturally mating to each other. So these are records of the attempting artificial insemination in dogs in early 1700s, but it was only from 1980s onwards that it became the commercially enterprise and became more widespread. Why is that? Let's have a think about what would be the issue with the artificial insemination. What would be the difficulties of that? And why couldn't we do like an ancient time? Why couldn't we do the 1700s? That's the first attempt. But why are we like overly widely spreading those techniques now? Think about what happened in the 1980s compared to 1980s, 1700s in terms of the technologies. Like what? Think about the sperm cells. It's actually as long when the sperms are ejected out, they started again dying out due to some unmatched environment issues. So you need to keep them in the ideal temperature, ideal humidity, or ideal somewhat like the environmental factors. Or at the time of the 1700s, they didn't have the machine called the refrigerator. We are now able to freezing. If you think about sperm, this is how we're using those to store your egg or the sperm in the sperm out the bank. They put it into the fridge, basically going into the really low temperature, going up to the minus 200 degrees Celsius sometimes. Okay, so completely freezing that cell's involvement, cell's movement. And what we do is we kind of defrost that and then put it into that. So we are now able to store those sperm cells better than the 1700s from 1980s. So that the development of those artificial insemination techniques getting much more improved from it. And think about from there. That's the time that we have kind of boomed all the different type of species. We introduced a new species into the biodiversity from then too. So one type of the artificial insemination is the animal husbandry, which means basically we take the sperms out. This is how we get the sperms out of the kettle, the sheep. Actually, like that. Okay. So the process involves that collection of the semens from the males and the insertion of the fresh or the stored sperm into the female's reproductive tracts under hygienic conditions as well. Something that's very important. The sperm can also be frozen using a technique known as the cryopreservation, where the biological samples are cooled to very low temperature, usually minus 135 degrees Celsius. That's extremely low the temperature, by the way. This allows the long-term storage and the transportation of the semen without losing the viability. Artificial insemination also allows for the impregnation of the multiple females animals in different geographic locations. Now, a good example is, do you know that we have the Wagyu in Australia? Okay, Wagyu is kind of originated from like in Japan. Now, yes, in the beginning time, Australian farmers kind of imported the Wagyu, the beef, not the beef, Wagyu, like the cow itself. But now that's time and cost. That's a lot of the time and cost. It means that they started now. What they do is they get the semens of the sperm cell of the Wagyus in Japan, put it into the Australian the cow or the kettles, and then now we have something in the mix of the Australian and the Wagyu beef. So this is how we get the Australian Wagyu, which is way cheaper than the Japanese Wagyu. So the Wagyu meat that you probably see around, you can commonly see in Australia, because Australia is one of the countries that we actually have the Wagyu beef, which is not the pure blood of the Wagyu in Japan, have some of that. That's a good example of the animal husbandry in Australia now. Same things happen like in some chickens and other animals as well. Okay, two, the good advantage of that is we can conserve the wildlife like a pandas, good example. We do them many times. So pandas, it's like they're about to extinct it due to there's some environmental issues too. In China, this is very unique like an species we only found in China mainland too. Do you know that? There is a panda like I call it some laboratory that's only looking for those. They're taking the sperm cell out of the pandas and put it into like some artificial like IVF, and they raise that number of the species now. So that's another example. So use the wildlife conservation to improve the reproductive success of the endangered species. So that's basically what happened in the zoos a lot. This is advantages as animals do not have to be transported a long distance between the wildlife centers, which can place a strain on the health and requires teams of the specialist animal handlers. Introduction of wild animals to our new potential mates can also result in the lack of the natural mating behavior of the even aggression leading to injury. That's basically the problem with the pandas too, because they aren't really willing to have a mating. I don't know, like that's basically lazy or like a lack of those, like some sexual desire from that too. So sometimes we need to do so. Third is another type of the reproductive technology is the in vitro fertilization, in short IVF. Now, how the IVF proceeds? First of all, basically you have some consultations. Okay, that's basically what happened. You get some samples. So ovarian simulate the ovarian cycle, so to get the mature like egg from the mother, the potential mother, or like a female. You also maturization. Then you get the egg from the mother. Okay, you take the egg out, and you get the sperm cell out from the father or like the male. And then what do? They're kind of mating them outside of the body first. Okay, we fertilize the egg. So IVF, highlight them here. IVF differs from the artificial insemination in that an egg is fertilized by the sperm outside. Highlight that place outside the mother's body. Artificial insemination means inside of the female, but this is outside. We go take both of the egg and sperm cell out, fertilizing outside of the body, and then kind of reinsert it into the mother's womb, or uterus in that case. So resulting zygotes are nurtured until they have progressed to early stage of the development. They are then transferred into the biological mother or surrogate mother or the frozen in liquid nitrogen for the later transplantation or for use in the scientific research. So this sometimes goes with the moed. Moed. What is a moed again? M-O-E-T. Multiple ovulation.
Embryo transfer.
Embryo. Embryo transportation. Means that potentially we can have that marry the female cat. This is basically not for the female, but not for the humans, by the way. Okay, that's mainly for like in the cattle, or mainly for like some used in agriculture too. We take the eggs out and fertilize the outside and put it into the multiple female cattle, not the humans. Okay, that'll be like some huge ethical issues with that. So that basically allows cows, which normally give the birth once a year, to become stud breeder when the surrogate mother are used. Direct embryo transfer is the final stage of the IVF, and then with the fresh or the frozen cultured embryo is transferred into the uterus. Insertion of the embryo is done using a thin tube called the catheter, which deposits the embryo into the uterus lining that too. So in many in Australia even now we have a little something to support for the IVF. That's something biologically somehow, like we don't know, like some males and females, they can't really have the babies, so they go for like options for the IVF too. This is quite common. A lot of people actually go for the IVF nowadays. Well, it is really. I've heard that like a per IVF, it's more than that. It's a
There's like a lot of people.
30K? No. It's I think it's like 10K per trials.
That it doesn't like it's not very necessarily successful.
No, it's so if you get it once, like you're saving lots of money. Some people go for like a 10 times or 20 times, but so it's cheaper in some of the countries, it's expensive in some of the other countries too. Now, good thing is IVF has some of the ethical issues or the religious issues too. So yeah, some people are actually going against the IVF rather than so this is something that we can think about it in terms of the social implication of those genetic technologies. So that's page 66 describing some of the advantage of the IVFs. You can read that through. That's basically extra information. So in the reproductive, we have artificial. Don't lose the track. Artificial insemination. We have the IVF or the moed. We also have the artificial pollination, and this is pretty common. This is actually really common, and in like some agricultures. Now, in like if you can do the same thing in your backyard. If you have the backyard, if you don't flowers, you don't get the flowers. You actually do that because nowadays, due to some climate change, we don't have many bees or the bugs. We learned about how the plants can go for the sexual reproductions too. The pollen needs to be embedded too, but sometimes we don't have a help. Like they don't have a help of that. In that case, they go for the process called the artificial pollinations. So highlight the first dot point, please. Artificial pollination occurs when the humans manually facilitate the natural plant's pollination process. The pollen is taken from the stamen with the male part of the one flower and dusted over the stigma. We learned that about the stamen or in the male and female parts. So resulting in the fertilization and then development of the seeds. These seeds may develop into fruits or the new plant, and those artificial pollination allows humans to choose pollen from plant with desired characteristics and to spread these genetic traits quickly through a population. So somewhat this is something very similar to the selective breeding, but pollination is the only term that we're using in the plants or the flowers. We don't really use the pollination in the animals. We rather use like fertilization and the artificial insemination or like some selective breeding in that case. Okay, now Mendel Do you remember the Mendel's? Yeah, Mendel actually used the artificial pollination to determine that, because think about his achievement, think about his in the scientific work. He used the pea plants, but he didn't wait for them just naturally going to matings. So what he has done, he chose a very specific phenotype, then two, like a red and the white flowers, made them kind of artificial pollinating to each other, seeing the next trait, and the generation that too. As we discussed in the theory of the evolution of genetics, that takes a huge many years. But using the techniques, he could have finished his work in his lifetime. Otherwise, if you just waited for natural time, using the natural pollinations, it's gonna take about more than 100 years for sure. Okay, so that's the example of artificial pollination now. So for question 1, could you define those selective breeding, artificial insemination, and in vitro fertilization, artificial pollination? Just writing down the definitions for now. Just one sentence would do. Don't forget to put the keywords for each of the definitions there. It's really important to have the good definition for all the keywords with the keywords. Okay, for the selective breeding, Kyuna, what would be the keywords to describing that selective breeding? Keywords, like a definition of the, or the definition there.
I said when humans selectively choose two animals, one with a desirable trait and the other with another desirable trait, and breed them together.
Good. So in the keywords, that would be desirable trait of the keywords, and we are selectively chose that. So it's basically a method of the breeding plants or the animals. It doesn't really relate to the limited the animals to produce to produce the offspring with some desirable, can't spell desirable, D-E-S-I-R, yep, D-E-S-I-R, desirable, the trait. So keyword describing this is, first of all, you are choosing the breeding or selective breeding, or choosing that for the desirable trait, for the next trait in the next generations. Good. What about artificial inseminations? Hannah, what would be the keywords in the definition of the artificial insemination then?
It occurs inside of the females. Like you take out the sperm.
Take out the sperm from male.
From the male.
Yep.
And you artificially put it into one or multiple females for the offspring to hold the desirable trait of them.
Yeah, it doesn't have to be the multiple like females, because the IVF we don't really do that for females. So it's the keywords you got to write. Okay, it's basically taking it from sperm, taking the sperms out. Sperm is introduced. Sperm introduced into the reproductive tract, the tract of the of a or many, sometimes one or many females without, this is important, without any sexual intercourse. Intercourse. This from here, this is the interest into the tract of the female from the sperm cell. So we get the sperm cell out and put it into the female's body, basically. Okay, any sort of so we had the example of the artificial inseminations. There was the husbandry or like some for the wildlife conservations. Remember that as the terms too. Okay, bit running out of space. C, IVF. IVF. Haley, what would be the key words describing the IVF or in vitro fertilization?
Fertilization and development of the zygote outside the body.
Good. Yes. So we get the fertilization, basically sperms and an egg. It's actually a sperm and an egg fertilized outside the body, and then introduce into the female reproductive tract. Let's say in laboratory settings, because we only able to do that in the labs, in the petri dish, basically. So the keyword would be outside, fertilizing outside there. So IVF or the MOAT is somewhat like a, overcoming the limitation of the artificial inseminations. Artificial insemination was overcoming the selective breeding, that you may or may not, there's a huge chance of randomness as time and costly, but this is more efficient in terms of time and cost-wise. It's actually better. This is even better too, if you can control that. What about D? We have that artificial pollinations. That's basically for the plants only, so artificial pollination is defined by the method of the transferring pollen from the male pollen to the flower. Transferring pollen to a female reproductive tract of a flower or the plant. Artificially, basically. So in other words, we can generate the new species using the artificial pollinations. You can mating that something from like flowers, like maybe like some similar features, carnations or like daisies, good example too. We can making the new flowers too, or making the different colors of that too. So question 2, what would be then some outcomes of the reproductive technology in the question 1? Let's talk about some outcomes first of all. As we're going through any of those techniques given in here, okay, there are some advantages, maybe like mainly for the advantages. Yes, of course, there could be a disadvantage, but let's talk about the advantage of using those genetic technology, which is reproductive technology first of all. The one thing, the benefit of using this is you are you're increasing some genetic diversity. Especially in the desirable trait. I mean, compared to like a few hundred years ago, we may have more new species compared to them. Okay, and then the new species coming in, introduced like a kind of every year, because we're working like a kind of commercial like in systems, means that you are able to create a new kind of species too. 2, another benefit of using this genetic technology is we can enhancing some fertility. Especially the IVF, think about the purpose of using the IVF in the humans, like that's would increase the fertility rate and kind of increasing or maintaining the number of the population. That's also very important. So you can kind of elaborate, you can just expand that from it. I just give it titles. 3, this will also improve, especially in the agricultures, improving the crop like in the yields. We kind of talked about that in your topic test last week. That's one of the questions. You can see it today is BT eggplant. Yeah, one of the eggplant, or some of the different type of like crops. They are genetically modified, or just kind of using the artificial like pollinations. In that case, we are just try to get this benefits or the good traits, strength of that. So these are known as the 3 some benefit outcomes of those, the reproductive, the technologies first of all. Then, of course, you can talk about increased number of the population. It may or may not, depends. Any questions about reproductive technologies in genetic technologies? I'm still doing that genetic technology, guys. Okay, reproductive technology, which is these are one part of the genetic technologies. Now, the second part of the genetic technologies, one of the part is the cloning. And cloning is something that very easy to understand. It's basically copy and paste. Copy and paste. Now, you have to understand that the cloning is somewhat very different to, have some similarities, but somewhat very different to the reproductive technologies. This is something that creating, or like something making the exact same feature. Like these 2 ships, they are identical, genetically, physically identical.
It's copy and paste. I think you did it. This can't be real.
Yeah, it's a copy and paste though, basically. It's not real.
So I was gonna say.
Too,
my eye.
yeah, too good to be exact.
That was amazing.
Okay, so just to highlight in here, we have that some cloning are discussed in the chapter. We have a gene cloning, highlight that please. We have a 2 type of cloning. We have a gene cloning. We also have the whole organism cloning, whole organism cloning. Sometimes like a whole like somatic cell cloning too. That's also describe that too. So gene cloning occurs at the cellular levels and involves producing identical copies of the one gene. Multiple copies of gene are needed for the genetic engineering and biotechnology research. But whole organism cloning, also known as a reproductive cloning, involves that creating a genetically identical organism using a somatic cells. This doesn't really involve with the sex cell, the gametes in this case. We just get from your hair, you can create yourself to identical Haley, identical Hannah, identical CUNA. Well, that's actually scary. And this is what having you.
What do you mean?
I'm just saying, having like another Haley would be with another Hannah, 4 of you here.
4 of us, 4 of me crying, yeah.
CUNA, I don't know, maybe. But 4 of you, well, that'd be a nightmare.
What? I don't know what you mean.
I can't imagine like how you're gonna deal with your own clone. All right, let's start on gene cloning, please. Gene cloning allows scientists to produce exact copies of the genes of the interest. So we are just selectively selecting the genes and we're copying that. It's very different from the cloning of the whole organism. In gene cloning, the end product is many copies of the specific genes. So 2 techniques, we use either be in vivo or in vitro. So in vivo techniques, methods, or in vitro. So in vivo means that use the something inside. In vitro means that we're using the restriction enzyme for the in vivo techniques, ligase and the vector. This is also known as we're going to learn more about this in the more details later on too. So what they do, the genetic code is universal and same, which is like, oh, it's a bit of typo, in all species and organisms. Yeah, I need to fix that. Now, in vitro gene cloning techniques, the polymerase chain reaction, also known as PCR, is used. Yes, we've been hearing about PCR in the COVID test. This is actually the same method. We're taking a gene and taking it out. So, the multiple copies of specific genes, and we learned that in chapter 6 pretty much. And chapter 6, gene cloning has many potential benefits for individuals there. I really need to edit this. This is something really terrible. Okay, so page 72 shows us some example of gene copying. This is also known as the recombination there. For the human cells, we're taking some genes that we're interested, and using that, bacteria cells plasmid. Okay, so remember what the plasmid is. There's a kind of ring-shaped DNA that I kind of describe that as kind of piggy bank. Do you remember this? The bacteria carry kind of piggy bank. So in that case, they need to repair that. They break that out and kind of fix that. So they carry with the lots of the bacteria, like some chromosomes. They carry the plasmid, which is kind of circular DNAs. What we do is we kind of cut the section of that and kind of attach the gene that you're interested in into that. We're going to learn more in details in techniques of the recombinant DNA later on too. But after you combine this, you can put it into another bacteria to kind of culturing that DNA. So you're going to copy and paste. Because what we know about the bacterial cell, guys, remember this? Bacterial cell is the one that in the prokaryotic, they are going for the asexual reproduction, which means they can go for any of those binary fissions. But the important thing is they can copy and paste of their own kind. What that means is you get some copy and paste, some copy of the recombinator one, put it into the one bacterial cell means that you get the copies of those kind of inherited too. So we are using some characteristics of the prokaryotic cells, asexual reproduction in this case. Okay, so this is for the in vivo. Now in vitro polymerase chains, page 73 shows how the PCR test is done. So what we do for the PCR test is basically exact same as your test you did, your COVID-19 test. You get to put it into the PCR in some of the samples, so in case it could be a bacteria there. And what we do is we put it into some of the high heat. They go denaturing. Basically, we have to separate the strands. You are copying those sections. We use in the primers, and then we use in the extension new strands to synthesize that too. So basically, I'm artificially copying those strands. And this is something that's very important in the PCR test, that you may see that some of the sections, this is the thing, virus can alter your DNA structures. I mean, that's what they do. We're going to learn that in the module 7. So once your cells or DNA get infected by the viruses, you may have some of the modification of some slight like a mutation of your DNAs. So what we do, from the PCR test, you remember you kind of shove it in, stick into your nose or mouth, Okay, put it into sample, and then they send the sample to the lab. Those lab people kind of put it in the kind of machine, these kind of machines, and then what they do is they're copying and paste of the DNA samples that you have. Then in that, they are able to see that some of the DNA strands are kind of modified compared to other DNA strands that you have. Does that make sense? Okay, because when you're collecting a sample, you're collecting like some samples from your nose, means that you're getting lots of the DNAs of you. Some of the DNAs get infected, means that by comparing the portion of the infected DNA by copying those or analyzing the DNA strains, you will be able to know that whether you're infected in the coronavirus or not. So that's how we can use the PCR for the test to detect the viruses. But this is also used for the kind of copying of the section of the DNA of yourself. So that's how we can use for the in vitro cloning. Page 74, please. Now, whole organism cloning means that we basically copy and paste of you, copy and paste of everything, your head to toe, not the section of the DNA, not the gene, yourself. And this has many applications in the areas of medicines and agriculture now. We're using that for only for medicine and agriculture. We don't really copy and paste. We can actually copy and paste humans, and now it's in technologies. We don't really do that now. So, whole organism cloning is used in many areas of scientific research where genetically identical test subjects are desired for reliable research outcomes in animal husbandry and in agriculture, where cloning enables desired traits to be passed on to many offspring in the method faster than traditional breeding. So what that means is, if you have something like some traits that you want to pass it on to the next generation, rather we rely on the breeding, you basically copy and paste of that, which is things that are considered to be efficient, because you don't have to waste of time because you like the trait, you just get to keep the trait as you pass on to the generation by generations. Okay. So especially in plants, we do that a lot. Not for the animals, more complicated, but plants, through the techniques such as the cutting and the grafting, has been used for both commercially and in the home gardens for centuries to create identical plants. Have you heard like you could cut and put it into, you get the new plant? That's basically cloning. Yes, even though you don't understand, you don't know that that's actually you're doing something genetic using the genetic technology. That is actually the whole organism cloning there. Yeah, we actually do that quite a lot. Okay, but guys, have a think about the difference between you get the seeds of the plant, you get the plant. That means that due to some sexual reproductions, the offspring must have some kind of complete different genetic structures to the previous one. There's a chance of mutation, maybe, I don't know. If you want to keep the traits, if you like the colors of flower, if you like the taste of the plant, okay, you gotta just cut and plant that so that we are able to get the same kind, which is known as whole organism cloning. Now, this involved in the animals is so more complicated. So, page 75, it shows you how we actually do it.
That's a cute.
Yeah, I found it.
I like it.
All right. Artificial embryo twinning is a technique that mimics the natural process, which leads to the identical twins where the embryo is split into two very early in the development process, and the both halves the embryo continues there. So what we do, the both individuals develop in the same fertility. We basically, in the embryo, you cut that in half, and then you put it into the halved embryo into the surrogate mother. Then that mother would now have identical twins.
Wait, so can you do this for humans as well? So
Technically, yes.
If you want twins.
Ethically, no.
Oh. So you can make twins just because you want twins.
But that twin, remember that, had exact genetic structure.
So you don't even know.
They're identical.
Oh, so
It's not like the twins like we have naturally, because the twins, trust me, identical twins, even though they're identical twins, they have so different characteristics because they are genetically different. They are sharing lots of the commonality because they're genetically similar, but they are completely different features. This means they have exact same. So this is what I'm saying. Practically, yes, we can do it, Ethically, we are not doing it. It's actually illegal doing this for humans in Australia.
How about in other countries then?
I don't know.
Like
I don't know.
other countries with other genetic
You never know. Like there are some bad scientists around, like in the whole world, and that happened in the wartime, especially.
Huh?
Yeah, they're doing lots of experiments. Okay, now that's for the one of the techniques for the whole organism cloning. Second is something that more common, known as a somatic cell nuclear transfer. In short, SCNT. SCNT. So we have the SCNT is a part of that. This is more common. It's more common, like a type of the whole organism cloning. It's more complicated. So what we do is we actually had that, and then we succeeded to copy one ship, which is called Dolly. I remember that in 1996. Dolly is the name of the ship. Scientists were able to copy the somatic cell of the ship, rearrange that, and then get a whole new copy of that ship. So that's what is given here. So in the each time a male is cloned, the SCNT process involved in the 3 animals: one that donates the nucleus, one that acts as an egg donor, and one that plays the role of the surrogate mother. So the page 76 diagram here shows you that example of that what we've done in 1996. This was shocking at the time, by the way. It was the first success of the like officially the first success of like getting the clone something. So what we have now, we have a belt like a Doris some ship. We get this some cells out, donor cells, remove the mammary gland, and donor cells coming out and then put it into the culture of the low nutrients, kind of keep them. Okay, then what we see there is we're seeing the donor cells kind of like going through that, and then you get the other cell from that adult black yeast, and then this goes like taking the somatic cells and the egg cells, and then fertilize that. It's not the sperm cell, by the way. This is something that's very interesting. We're not putting the sperm into the egg cell. If you put in the sperm into the egg cell, that'll be one of the artificial insemination or the IVF. This is something very special because we are able to get this exact copy from the somatic cell, which is body cells, not a sex cell involved. So what they do, they remove the nucleus of the egg. What we need is, we need the environment of the egg cell, which can be divided, it might go to an infinite number of the mitosis, so that you remove the excess nucleus and then put the somatic cells of the ship into that kind of covers of the egg cell. So you don't really need to go for the fertilization. This will already be it comes in the diploid number of the chromosome, which is identical to this ship. Does that make sense? Okay. Then this goes into surrogate mother, and we get the Dolly, who is the exact same copy of that, genetically same copy of that ship, this ship here. So we're still using in that time at the time they're still using the surrogate mother, which is a female reproductive tract. We're still using the some egg cell. But you have to notice that the main differences between that SCNT to IVF. Okay, IVF, again, we put the sperm into the egg and make them to fertilized, and then put it back into the surrogate mother. SCNT, the somatic cell, the technology is you're taking somatic cells and one egg cell, remove the nucleus of the egg cell, put those somatic cell into the egg cell's environment, put it into surrogate mother, and get the exact copy. Okay? Can you spot the difference between the cloning to this IVF or the artificial inseminations? Again, artificial insemination means that you may have a chance like a different phenotype coming in the next generation, because we don't know. There's a chance. Maybe like you want to get the tender beef, but it's failed. Like basically, it's not showing you're having any tender beef anymore. But using the SCNT, if you think about just the pros of that, you always get the same delicate, juicy beef the same taste all the time, because that's a cloning, copy and paste of what you had before. All right. At the time, this is something that very special. And after this, 1996, after this paper is released, people were so frightened, because technically, we are now able to copy and paste yourself. We only need one of your cells, some cells, somatic cells. You don't need to take, means that we can copy and paste the females, not only the males. We don't need the help of sperm cells anymore. We just need any of those, whether that's a male or female. We just need a somatic cells of any of the species. Yes, means that you're gonna have you.
Generations of me.
I mean, not the generation. It's just you. It'll be you after you.
I don't want to me another me.
It's hard to call your daughter biologically, because that's basically genetically it's you. Okay?
It's me.
All right, I'd like you to answer the question 1, question 2, 3, please.
It's cold.
Sorry, I can't. It's better than hot. Yeah.
Oh my gosh.
The question again, whole organism cloning. So how do you define that? This involves that basically replication or simply copy and paste of the whole organism. We're taking the copy of the whole thing. This includes that, as we know, including that example is SCNT, the somatic. It's good to know the full name. Somatic cell nucleus transfer is something that we actually believe that more like common whole organism cloning. If you need to clone something, so what we do is we generally like the nucleus of a somatic cell. That's in the body is transferred. Into the write this one down into an in EUC in uncleated. This is how to pronounce actually. In enucleated, or like a kind of empty egg. Enucleated egg cell, which means basically this means that the egg cell with its nucleus removed. Okay, that's the whole organism cloning. And we got to talk about the steps of the whole organism cloning after the question 3. For the gene cloning is somewhat different to the whole organism cloning. We're not copying and paste the whole organism. In that case, the gene cloning is also this is also known as the molecular cloning, or the DNA cloning too. We kind of mix use that. And replication of, we're not copying the whole thing, a specific gene or the DNA sequence of the interest. Okay, so techniques we're using this for. We commonly use the PCR, which is also known as polymerase the chain reaction. It's a techniques that we use quite a lot, or sometimes all the restricted enzyme is somewhat we use to kind of cloning process. We're going to discuss more about those techniques. Later on, this is also related to the recombinant DNA. We're going to discuss that further in the next chapter today. And this is something that's very important. You actually need to memorize the process of the recombinant DNA process too. And sometimes you need to know the process of the whole organism's cloning, which is SCNT. Interest. Question 2. Outline the process used to clone the whole organisms. So basically this is described well on the page 76. It's given here like that. First of all, the cells were taken from the other, which is the mammary gland of the 6-year-old eel. So basically we are taking the cells from the one sheep. Okay, well that doesn't need to be, that shouldn't be actually, that shouldn't be the sex cell. That's a somatic cell. Any of the body cells. And these cells were starved in the nutrients to stop them dividing. So this is why we put it into a low nutrient environment. Second, the nucleus was removed from the healthy unfertilized egg from another sheep and process called the inoculation. Highlight this one too. Enucleations. Then the cell with the nucleus from the this sheep is put it into the egg. I kind of call it like an empty shell, or the egg cell, without the nucleus. So put it into the inoculators, the egg cell, and then put that into the surrogate mother. All right, and then you get the exact same cloned sheep out of it. And this is actually really extremely hard because one, think about the process of taking these the cells, somatic cells out. As soon as you're taking the cells out, they start to actually get divided, going through the mitosis. So that needs to be really rapid. So this is why we're taking the many somatic cells, not just the one. So we choose the one that is not really going for any of the mitosis. Think about the process, guys. Think about the process of mitosis. As soon as they started going to mitosis, that means that they have the extra copies of DNA going through the DNA, the replications. This means that if that goes to fertilize further, there could be some mutated kind of genes. Well, maybe like there are some uncompleted DNA comes there. If they're successful to give the birth to the baby sheep, means that the baby sheep would have the 2 heads, 5 legs, 6 legs, because they have multiple DNA strands there, extra copies of that too. So this is really important that if you're going to get the whole organism copy, you need to find this cell without any mitosis. 2, it is extremely hard to extract the nucleus only from the egg cell. And think about how many egg cells produced per the sheep per month. So you actually need to get a lot of samples of the egg cell as well too. Because trial and error, you will lose some of the egg cells. You may destroy the cell structures while you're taking the nucleus out. 3, sometimes that egg cell with the somatic cells nucleus will not be accepted by the surrogate mother's reproductive system. And that's one of the issues too. So it took a really long period of time to go for that this kind of experiment. They actually had a lot of trial and errors. But after they succeeded that, we now have a few different type of clones, kettles. We have a kettle's pig. I remember that. Mice is kind of common. So we're now able to get the cloning the different type of the animals. Yes, practically we can make a clone of the humans with the same techniques, but we just don't do it. We just don't do it. Does that make sense to all? Okay, that's the process of the SCNT, or whole organism cloning then. Now, the reason why I'm not more talking about that artificial embryo twinning is because this is something what we don't really go for a lot. That has a chance of failure too. But this is one type of the whole organism cloning as well too. Does that make sense? And by the way, putting the embryo into a roller coaster doesn't mean that they're divided into two. This might need to go into the lab, and Petri dish, we kind of divide that. That again, we may need to provide the two empty inoculated like egg cells. Put the embryo into half and put it there. But this means that there are chances of you fail that get the clone. Okay, you never know. 3, explain the purpose of the whole organism cloning. Why we do that then? Why are we doing this?
It's more
Efficient in...
because you can get the desired traits without like
Yep.
possibility.
Good. So let's write it down. 1. We're the main purpose of doing this is, first of all, is for the biomedical research. Yet we haven't got having cloning means that we are able to consume their meat or get some benefit from yet. Okay, we don't really get that yet. So in many cases, we're doing for the whole organism, like cloning is for the biomedical researchers too. You're copying some of the parts of the body, maybe like research that as well too. 2, we also do for the this kind of inventing the reproductive, the medicine, and this again will lead into the agriculture. Some involvements, I think, improvements. Write it down. And as we can copy and paste, this is actually something happening in have you guys heard about the Tasmanian tiger? We have a sample of that, okay? Which means we'll probably be able to conserve, because they're extincted, we can maybe revive those extincted species as well too. So that's for the species conservations. Again, for the dodo bird again too, we have an extended dodo bird. We may just gonna get the some copy of the dodo birds. Maybe if you ever watched the movie called like Jurassic Park, that's basically showing you some example of cloning as well too. We get a DNA sample from like the blood of the yes.
Dinosaurs in my house.
Not your house. Okay.
You are the dinosaur in your house.
So basically, these are the main purposes. It's also helping. It's all like a research. So it's either be for the research purposes, producing some drug and medications, agriculture, and maybe some species of conservation for now. Okay, don't just limit the example to these cattle or cattle or the sheep. We may be like improving some of the clonings in plants, crops. We actually use that quite a lot. It doesn't have to be SCNT, by the way. Basically, cutting the plant and they planted it again. You're basically cloning the whole organism as itself too. Does that make sense? Okay, so we've done 2 of the genetic technologies. One is about, again, the reproductive technologies. 2, the cloning part. What you need to know is you should know the main difference between these 2 and some examples of those as well too. After we have a break, we're going to go through this recombinant DNA technologies then. The 3rd genetic technology, just video. DNA technology. But this is actually just some portion of the gene cloning too. We're using this recombinant DNA technology for the cloning the genes. This is something that we use a lot, and we're using this to produce a GMO, the genetic modified organism as well too. So what is a recombinant? Yes, as we can get from the title recombinant here, we are getting some recombination of the DNA. So the things you need to know about the recombinant DNA is, first, you need to know the step-by-step of the recombinant DNA process, because we actually had a HSC question that asking you, describing the steps of the process of the recombinant DNA technologies. So first thing, the steps of recombinant DNA technology is shown on the page 80. First of all, the two pieces of DNA are cut using the same restriction enzyme. It's better shown, like you see that picture, the page 81. That's actually a good picture that I've got. It's from textbook. You probably can see that again too. So what we first do is we're getting some circular plasmid DNA. Remember the plasmid is, I described that as kind of piggy bank of the bacteria. We get the kind of the plasmid, which is circular DNAs from the one of the bacteria there. You cut it using the restriction enzyme. So that's basically working as scissors. Restriction enzyme.
It's just called restriction.
It's called the restriction enzyme. It is called yeah. So restriction enzyme is that enzyme that cut that some portion of that plasmid. Then what we produce from here is this site over here, which now becomes to like in a kind of open in the circular path. Using the same restriction enzymes, we also cutting some of the DNA section that you desire to get. So we already have researched that. One of the section is that what we want to get in terms of phenotype. We are all producing that kind of proteins. Then after you cutting that sections over here, this gene fragment, highlight this one please, are having some the matching sticky ends. How do the sticky ends? We're kind of cutting this into this kind of shape, leaving that. So these are the sticky ends. And as you can get from the word sticky, this base pairing pads on the sequence of base pairing will be matching to the plasmid base pairing as well too. So you design it to cut that portion so that the base pairing will not go wrong. We actually can calculate, can design to get this kind of sticky end all the times. Now what happened is you kind of put it into that this and some section of the gene into the plasmid, and using the ligase. DNA ligase, which is what? The enzyme that's helping to seal, like working as a kind of glue these one out. It's a little bit of engineering, kind of like it's a genetic engineering required for this. What you do after that, this is now called the recombinant DNA, because this is now recombined with the new DNA strands. Then you put it into the one bacteria, there. As we know, the bacteria can go through what? The asexual reproduction, which is basically copying and pasting on itself. After leaving that in the grown agar or like in the culture, you're able to get that exact same identical copies of those plasmids. This is how we can get the gene cloning, and the whole process is called that recombinant DNA technologies. It is very important to know that the process of that producing the recombinant DNAs, or how do we get that in each of steps. So make sure remember this, memorize this by next week. You will get at least one question related to this. You will actually get the process question next week too. You actually have that in your HSA question, your homework. So make sure you try that. So page 82, please. Let's go more in details about those step-by-steps. So we're using the plasmids, something known as vectors. Vectors. Now, vector is not a vector that we are learning in mathematics. Vector in molecular biology means that it's basically guiding something. We have a guidance of that. So that's basically is DNA molecules. Vector is the DNA molecules used to transfer the foreign DNA. Foreign DNA into a host organism. So this is basic, because think about it here. Even though we want to copy some section of the genes, you're not actually able to copy them that exact same informations. That's not really possible in our technologies, like artificially we can copy that. So this is why we get the help of the bacteria, because we know that the characteristic of the asexual reproduction. And the vector, there are different type of vectors we can use in organic, in molecular biology, but we just gonna focus on the plasmid, which is the what? The DNA, circular DNA, which you can find in the bacteria or the prokaryotics. Okay, now this is something that we commonly use to copy and paste of the insulin. That all the insulin that we need to have that, okay, in the genes. So insulin is basically what? Hannah, what is insulin?
It's it has to do with sugar.
It has to do with the sugar. It lowers the sugar levels too. And you know that we can inject the insulin as an artificial insulin. Think about it here. You can't draw out insulin all the times to inject to someone else's. But somehow we have a fast-acting, some of the long-acting insulins kind of in the pharmacy we can buy. That's basically made with this process over here, using that recombinant DNA the technologies. That specifically we're using the plasmid of the bacteria to copy and paste the insulin informations, pass it on that, so that we get the manufactured insulin, so which you can inject it into the patient with the diabetes. So that's given here. For example, if a recombinant plasmid containing the insulin-coding gene is placed in the bacterial cell, the cell would produce a large amount of the protein insulin, which can be used in the treatment of the diabetes. So plasmids are used as vectors as they are self-replicating. Highlight that please. It is very important to know that this is the why we chose a plasmid or the vector. Stable and easy to manipulate in the laboratory. This is something that's very important that we have in the lab. So they are vary in size and functionality, but contains several common features. One, the origin of the replication, also called the ORI. Okay, at least one restriction enzyme recognized site, and a selector markers or the reporter genes there. It's actually like a bit of like depth study even here. They were probably not gonna learn up to this point, but this is important to know. Or use your common sense. Okay, that makes sense that why we choose the plasmid as a vector. One is that's it can replicate. One, it has a site of the acting for the we can cut. In other words, we can cut that. Restriction enzyme. Remember that that's a cutting, like a scissors. We can find the cutting point. They kind of marked. 3, the selected markers or reporter genes there. So this is how we can show like some of the sections over here, and that's the section we are able to cut through this, which means that we can stick the new genes into the plasmid then. Next point is we are creating the recombinant plasmid using the vector or the plasmid in this case. So in the DNAs, you get the after you're cutting using the restriction DNA, you're making the section called the sticky part or sticky ends. The sticky ends is where we get the base pairing comes in that with the very specific in the base order. So that is matching to the cutted that plasmid too. So what they do, they comes together first of all, and then you use the ligase to glue them out. Does that make sense? So this is very important that we, when you're thinking about the kind of cutting and pasting something, you need to cut in the same identical like a shape, so that when you just place them, when you glue them, they comes as the one. And this is something very important in molecular biology because we need to have the whole plasmid. Otherwise, bacteria will recognize that that's not a complete plasmid. They will not really go for the asexual reproductions. This means this needs to be taken really big, concise and really long period of time taken for that, but something that's very efficient once you get it. After that, you're transforming bacterial cells, put it into that new bacterial cells to make sure they go for the reproductions so that we can copy and paste of that desired DNAs too. So it looks really complicated if you haven't seen that before, but once you understand that what we do, it's pretty simple, the step-by-step guideline. So let's summarize that. That's basically selection and screen the transform bacteria. That's the further information. And using these techniques, we are able to create or produce something called the genetically modified organism. We discussed that last week and the week before a lot, the GMO foods. So both bottom here, corns, peas, potatoes, melons, apple, papayas. Papaya in Hawaii, do you remember that examples? There's a genetically modified as well too. These are known as the most popular genetically modified the plants or the organisms too. It doesn't mean they're all bad. I mean, it doesn't mean like, consuming these you get like some genetically modified something in your body, because they're going through this process of the recombinant techniques there. Does that make sense? Okay, it doesn't mean like it can cause you to get some mutation, get a cancer from it, because there was a myth about it before. So this pros and cons, we kind of talked about it last time, so I would just kind of skip that and just put some extra information in here for now. Any questions about GMOs for now? No? All right, next page. Highlight the place on page 87, transgenic species. The GMO and transgenic species is somewhat a little bit different too. So GMO is defined by we are genetically modifying the code. Actually, from the previous page, highlight this one please. An organism that has the gene altered in this way is considered to be a genetically modified the organisms. So what we do, the desirable characteristics seen in the one animal or plant could be transferred to another organism lacking this characteristic. So we're borrowing for others genes, which has probably having a strength or the benefit, and implementing into the new species there. Like potato is a good example, or the tomato is a good example, or even the banana is a good example too. Probably told you that in an ancient banana wasn't like the bananas that we're eating now. It looked like a melon at the time. Now what we get in the kind of juicy, kind of soft banana is because we are implementing some other plants. And you've probably seen that some like grapes these days, kind of sweet grapes. What was that, like a grape? Yeah, the cotton candies. Yeah, that's actually kind of genetically modified organisms too. Because we are borrowing some of the sugar part, the sweet part, kind of crunchy part of the genes from the other like fruits. Kind of combination with the apples probably, and they're making that into some grapes, but tastes like a bit of like apple features, like a sweet and kind of crunchy there.
But do crispy grapes love you?
I have crispy grapes.
They have no emotion, so they will love them. Okay. All right. Transgenic species, highlight that please. Gene manipulation, known as genetic engineering, can be used to create the transgenic species. So you have to know the difference between the GMOs, the transgenic species. This is genetic gene manipulations or gene engineering. It means that transgenic species is one that has been created by moving gene across species, taking a gene from one species and inserting into the DNA of another species. This gene must become part of that organism's germline genome with DNA if it is to be inherited by the subsequent generations. This gene is therefore inserted either directly into the germline cell or into the fertilized egg cell. The creation of the transgenic species is only considered to be reproductive technologies if it increases the reproductive capacity of the animals. And most transgenic species are created for the reasons other than increasing the reproductions. So a good example is, we talked about the BT, like eggplant last time, but BT cotton is something that we commonly use for the transgenic. The BT stands for like a bacteria, by the way, like it's the type of bacteria. So what we do is the bacteria BT cottons, the second last point please, were genetically modified. This means that added or removed the genes. They contain the gene that codes for the production of the protein that kills the caterpillars or the Helicobacter disease moth. So the insertion of the BT gene into the cotton plant has reduced the need to use the pesticides to kill these caterpillars. This is better for the environment and to reduce the development of the pesticides resistance in the caterpillars. The gene is called the BT because it was originally taken from the soil bacterium, Bacillus thermogenesis. So it's not only for the cottons, we actually get the inside in the BT, the bacteria genes into the different the species too. Like we talked about the eggplant. It can be like a banana. It can be it was a papaya. I remember that it was one of the examples too. If you don't have the diversity, remember that genetic diversity means that having a one selective pressure can kill everything. So it's good to have this. It's good to have that. So cotton guru is that the most of the cottons you're wearing now, even this are made with the BT cottons in that reason. So it's somehow environmentally safe, but issue with that. Okay, let's talk about a bit briefly about the ethical issues we use in the BT cottons. Haley, what about the issue with using those genetically modified or the transgenic species then?
Nothing.
There would be something. Ethical issue means that you got to think about one, whether there are benefit and harmfulness. Two, fairness. Right, but basically fairness. The well-being, fairness, right? Privacy? Was it privacy? It wasn't privacy. It was something really privacy, but in a while. Let's have a look at that again. Ethical, the principles. We told a choice. So we did describe that as WCRF. World Cup round final. Okay, World Cup round final. Wellness, choice, right, respect, and the fairness there. So in terms of using the BT cottons, yes, wellness. Let's talk about well-being. It is really good that we don't have to deal with all the environmental problem issues. And wearing the BT cotton, the cloth, doesn't mean that you get any genetic disorder. So in terms of the well-being, that's good. What about the choice? Choice. What the choice stands for? Have the freedom of choice. Do you think the farmers now having freedom of choice? They have now, but think about the farmer's perspective in there. If you have BT cotton, which you need to use the pesticides anymore, have some cottons, but this comes with the fairness as well too, or the equity. BT cottons are known for more expensive. And the problem with some of the BT is they do not really inherit in the next generation. They don't really go for the, so it means that they need to buy the new seeds from the company, in many times, to have the BT cottons again. This means what? There will be rich people get a reach out means it comes with some ethical issue called the fairness. It's not the proper or a fair distribution of the equity of the assets in that case. What about the respect? What about respect? It might be some respectful too in terms of the environmental issues too. Remember that World Cup round final. World Cup round final. Okay, to understand the ethical issue, because you may get the ethical issue questions in your trials. Okay, and your HSC this year. So let's get prepared for that. World Cup round final. Remember this. Going back to transgenic species, this is the BT cotton shown on your page 88. This is kind of case study showing you that. So it's in terms of it's good. So the process used to produce transgenic cotton is follows. It shows you in here. Scientists cut the normal cotton seeding into the small pieces and place them into solid growth medium where they grow into calyces. After about 6 weeks, the callus cell will transfer to a liquid medium where they are given hormones to induce their growth into cotton plant embryos. 2. By genetically engineering, the BT gene is cut from the genome of the bacterium. Okay, and then put it in using the restriction enzyme. So basically using, again, the recombinant DNA technologies for this. The BT gene is transferred to the cotton plant embryos. This is done using the second bacterium as a carrier of the carrier or vector. So in this case, we do not use the plasmid, we're using the bacterium as the whole the vector in this case. This bacterium called the agrobacterium tumefensiums causes the crown gall disease in the plant, and it's used as a vector because it's able to inject the genes into the other cells. So sometimes we actually do that. We actually sometimes use the viruses for the vectors because they can migrate that from one to another quite easily too. For the cotton plant, embryos are dipped into a solution that contains a mixture of the vector and this bacteria, and they extract the Bt genes and the vector bacterium inject the Bt gene into the cotton cells. Then what happened? We now get, once the gene is inserted, the embryos containing the Bt genes are grown in the tissue culture and they're placed on another solid medium and are germinated into small plants which are placed in a pot and grow in a glass house. The plants are now a transgenic species containing a gene from another species in their genomes. Does that make sense? Okay, so a lot of times we're using those the benefits or characteristics of the bacteria or some prokaryotics to create that. Then how to use those transgenic organisms in the medicines? In medicines, we're using the many we're using the kind of transgenic animals. So highlight the second point, please. The transgenic animals can be used to study how genes regulate specific body functions. And this is kind of recent, relatively recent study, like in the 2007 Nobel Prize one. Okay, that we use a knockout mouse. The knockout mouse is the one in which researchers have inactivated or knocked out existing genes. So what they do, this knocked out gene has been replaced with an artificial piece of DNA at the blastocyst stage of development. Means transgenic mice with the gonads that have the knockout stem cells are crossbred, producing some offspring that are pure breeding for the knockout genes. These are used for the research and the making the specific gene inactive and observing the difference between from the normal behavior of the functioning. Researchers can infer the function of that genes. So what we do is we can't really do a human studies though, but mice is kind of something that have a really similar the DNA structure as the humans. So they are artificially, purposely deactivating some section of the genes of the mice. Then, if that shows the mice to be like some dysfunctioning in some or like having some difficulties or something, you are able to know that, oh, that gene is corresponding to that motion, or that gene is corresponding to skin color. That gene goes to maybe the IQ, maybe the brain functionality. So that is how we use in the medical researches too, because mice and humans, as I said, have a very similar characteristic of the sequence of the DNAs. This can be applied to humans as well, but as we are, by looking at some genetic diseases, guys, okay, means that we are able to know that which part of the genetic code is wrong, which means you're able to research the part, there's a chance that we can cure their genetic diseases too. Does that make sense? Okay, so let's define the following terms in the recombinant DNA technologies. Let's start from question 1. Recombinant, the DNA. So what is recombinant DNA? What are the key words for that? This is the DNA molecules that are formed by the joining together DNA sequence from the joining together the DNA sequence from different sources, from different species or the sources. It doesn't have to be species, but for example, from the previous medical examples, to deactivate that, we don't really get the sequence from the organisms. We are artificially making that, which is making the deactivated forms. Okay, that's how we can define the recombinant DNA. It's all about the DNA molecules. It's a DNA molecules that's recombinated or reproduced. What about sticky ends? Sticky ends. Or end, I should say. Sticky end is a single strand. Stranded. The in terms of the overhang that is created when a DNA molecule is cut by a restriction, the enzyme. Do you remember, like, in some TTAA part of the AA, like this, from the gene? Okay, so TTT, so or TTA, doesn't really matter for the order. This is called a sticky end then, which is like a single strand that will be able to stick to the plasmid or the vectors later on. All right, what about a transgenic organism? Transgenic organism. A transgenic organism is organism that has been genetically modified. This is the organism that has been genetically modified to contain the DNA from the different species. Okay, let's write it this one too. The foreign DNA is known as a transgene in that case. Okay, vector, as we described the vector in organic molecular biology is that the DNA molecules is a DNA molecule and used to transfer the foreign DNA, which is transgene into a host organisms. That's not spelling. Organism. That's called a vector then. Lastly, plasmid. Let's redefine the plasmid again. Plasmid is something that we can find in the bacteria. It's a small circular DNA molecule, a small circular DNA molecule that is separated from the basically in the prokaryotes, which contains the extra information of the DNAs, and don't worry about the genetic engineering. Basically, that's basically manipulating genetic materials. That's how we can define that. 2. Use a diagram to explain how a transgenic organisms can be created. This is just simply asking you show that diagram that we discussed before. That's shown on the page again. 81. This is the diagram you can use. That started from everything from the plasmid as a vector, cutting that plasmid using the restriction enzyme, making that producing sticky ends. Okay, we do the same. We select. So let's talk about the steps first over here. The steps of the DNA recombination? Or recombinant DNA? It started from one here, selection. So you're selecting the very specific gene that you decide to want to get. Like it depends like what kind of like a characteristic you want to carry. So yeah, select is the first process that we do. Two, after that you're cutting. Cutting both of the plasmid, cutting the vector, as well as the gene, and producing, cutting, and the sticky end are now produced. To cut that vector and genes, we're using the enzyme called the restriction enzyme. Does that make sense? The second part. 3. After you cut, you pasting. You paste. Just say paste. Cut and paste. The paste that the gene, which is with the sticky end part, to the vector sticky end part. Okay, and then what do you do after? After that, you transfer into the new bacterium. After that, you culture. You're gonna get the more copies of that, and then utilize after this. So how many steps in general? We have 5 steps of the DNA recombinant DNA technologies. Selection of the right gene sections, cutting the vectors and genes using the restriction enzyme, and producing some sticky ends for both. Paste that, and to completely paste that, we need to use a glue called the DNA ligands in that case, and then you transfer into the new bacterium cells. Then you culture that to make that more copies of the plasmid or the DNA recombinant DNA in that case. So in short, you may just memorize that as these the 5 steps, or just get the synonym like SCPTC, if that helps you too. Okay, that's how we can describe about the recombinant DNA technologies then. Make sense? Not too bad? All right, next question please. Outline the recombinant DNA procedures and use the yeah, basically that's what I did in here. Insertion of vectors, and yeah, that's basically what we talked about too. So let's look at those. Okay, skip this question for now. Let's look at those like in genetic technologies then in the medical benefits. Now, I'm not simply talking about just the recombinant DNA. We're not talking about genetic technologies. Guys, everything on the board in here, there are the genetic technologies. So let's discuss about some benefits of using those genetic technology in medicines. I mean, we've been talking about it quite a lot. It can be any of those. So first of all, in the medicine, what we can tell is it's something that we can use for the, improving the medications, improving the diagnoses. So artificial, for example, insemination which is mostly via intrauterine insemination, allow human to conceive a child despite challenging, including the fertility problem of the absence of the sexual partner. In other words, yes, IVF could be one of the options too, but it doesn't need to be the IVF all the time. Sometimes we get the surrogate mother. You know, that things happen. Yeah, we get surrogate mother to get the biological, like the child. Or cloning holds many potential applications for the medicines too, as we discussed too. One biomedical application is xenotransplantation, which involves the transplantation of the cells, tissues, organs from the one donor species to humans. So what that means is, guys, this is possible. Assume that you have some damage in on your body parts. Say you got blind due to the accident. Then what they do, you take one of the body cells, somatic cells, cultivate that, making the stem cells, or copy and paste the informations, or you can just kind of activate the portion, the section that's making only corresponding to the eyes, make that a new eye from it. This is what we need to know about the mapping. This is called the genetic mapping. If you know like a section of that section is about the eye part, you deactivate the other parts. You only activate the eye means that cell will now cultivate it into eyeballs. So you get your own eyeball again implemented. So technically, you can live forever.
Yes. No.
As in like, you can replace your body part. If you get something wrong, you just replace that. If you get some joint problem, replace it with a new joint of yours. Okay, just like a robot, you're replacing everything, like a parts of the bodies. But that's not happening yet, and that carries lots of the ethical issues, especially in fairness. So those rich people can afford to pay for that, all the research and that, but others, they may not able to. Okay, cloning can produce also the genetically altered cell that can be used in treatment of diseases such as Parkinson and Alzheimer's. Okay, highlight this. Parkinson and Alzheimer's used to be like a non-curable. Now, because you can make a clone of that, you can replace the cells, you can cure that Parkinson and Alzheimer's disease. There's not a perfect cure, but you can improve that symptoms. So it is helping us. It is very like precise that in terms of the targeting the very specific genes too. What about the recombinant proteins that produce the recombinant DNA into the bacteria? So that what we do, there are hormones are produced from that too. So not only for the insulin, we talked about the insulin. Yes, we are using the recombinant DNA technology. We are producing huge amount of the insulin. Guys, what about this? If we have a female who has some problem with the gland, whether that be pituitary gland or some ovaries, so she's not able to producing the certain amount of the oxytocin or the female hormones. Which means technically we can copy and paste making lots of female hormones, which we do now, which is estrogen. You can inject the estrogen to replace the amount of hormones you've lost. Testosterone, have you guys heard that? Or steroids? Yes, that's basically made with the same manner too. And steroid is not only for you to do the muscle growth. If you ever had your asthma, if you ever had the Ventolin, yes, that's basically made with the same technique of recombinant DNA procedure too. The asthma puffer. So it's saving millions of people, tons of people using that, including my kids as well too.
Do your kids have asthma?
Oh yeah, every kid has asthma sometimes. So that's basically how we're copying that steroid, which is the hormones again, and we put in.
So we're putting hormones into our body.
It's a hormone, yeah. Like steroids, basically. Asthma or like some of the Ventolin, some type of the inhalers are steroids. So what they do, they're opening up the airway. Yes, because what happens in asthma is kind of closed airway, it's making them really feel closed so that you can't breathe properly. So by taking the inhaler, you're basically putting some hormones in it, then kind of opening it up. That hormone is made with the recombinant DNA technologies too. Okay, so lots of those too. Now, what about the agriculture? So page 96 shows you that some contribution of biotechnology crops, and that's 2D. One, that increases the crop productivity. That we have a less plowing, less of the some fertilizers using due to that, which is good for the environment. Two, helps to conserve the biodiversity. Yes. We actually discussed about issues with the biodiversity using the GMOs, but that's one issue. Three, reduce the agricultural eco footprint, which is like reducing all the environmental problem, kind of reducing it, not eliminating it. Four, mitigate the climate change eventually, which is good for the agricultures that we're using many benefits in that reason. So But we've been talking about lots of those some transgenic. Now, industrial benefits. What would be the benefit in the industry then?
Let's have a look.
Enzymes. Have a good think about here. It shows you that some use of the biotechnology we're using for some first foods, making them the biofuels too. So that's replacing that some of the crude oil with the fossil fuels. Okay, enzymes, having lots of enzymes. It's not only for it's good for the mass food production. Okay, when do you think we are able to get those in the East? A lot. Okay, that's also known as the bio or genetic technology. We are able to copy and paste of that of the East, so that you're not. If you want to buy the East in the supermarkets now, they cost like a few dollars only. Means you got lots of those enzymes, or like in the yeast, which the bacteria they can do. So lots of the E. coli is a kind of common like bacteria we use in the laboratory that for the culturing them and kind of investigating them too, some metals and some other things too. Okay, so what I'd like to do is I would like to answer these two questions at the back, question 1 and 2, before you go home today.